Any thoughts to add about the different ways that one could go about the development atlas, whether it's organ-based or anatomical unit-based?
So I'm actually wondering, because I mean, even though we have access to the fetal resource, which you work with, which is amazing, truly amazing, I think it's going to be hard to get some of the earliest steps. And if we really want to go from the very beginning, I always thought it would be a nice idea to try and, you know, get some of the earliest steps, which will be much harder to do in human, from a non-human with some, you know, orthology mapping, and, you know, map the human so that we can get some deep profiling where it's just going to be really hard to do in a good way from human, as, you know, we have the developing embryo. And then once we get to stages where we have much more data, then of course we can switch to pure human, as well as, you know, at that point the size will require working on organs separately. But I sort of imagine this timeline where we have this morphed model organism, human morphed, to get enough data for those early stages, and to have, you know, whole, you know, single animal map. And as the organs begin to become organs, then we can break off into more organ-centric atlases.
And can I just ask, I mean, it seems like getting, as Dana pointed out, getting those early, some of the intermediate time points in human is actually particularly challenging, just in terms of being able to access the material. So it seems like this would be, if somebody actually has access to it, to be able to profile all the tissues at once, it would be a big benefit. But I don't know how practical that is. So how far can we take it in terms of profiling the entire embryo?
You mean the beginning or the end of that period?
The beginning.
Yeah, but what do you? But what are you? So what do you mean by the? What is the beginning for you here? How many weeks are we talking about?
Well, let's say I guess I'm mostly concerned like within like week 4 or something like this, or week 5.
So I think there's the pre-implantation cultures that go up to 14 days, and then you can't really culture beyond that if it's a human embryo. And there's quite good data up to 14 days at the moment, you know, but there's a lot more that can be done still. Then you have like a embryonic stem cell or some other kind of culture that can go beyond that. And then there's a gap, obviously. And then what we can realistically get is really from, you know, 4 post-conception weeks onwards, because before that and the end of the 12th.
Probably had to use models, primates models, for detailed development stage, and until the you know, postnatal day 1, I guess you can have abortions maybe added, but precise time course has to be from models.
Yeah, so I think
and I think it would be really great to have some type of combination, because I think we can get really sparse data in some of the earlier points, which, you know, help get some type of alignment. So I think it would be pretty cool also to, you know, I'm as a computational biologist, I think it would be, you know, really cool to think about, I mean, both the data collection challenge of it, but also the computational challenge of it, of having a temporal map of a spatial temporal map of development, where you have higher resolution in some model organisms, but much lower resolution, some sporadic, you know, lucky samples from humans, and how to sort of merge that to sort of fill in the details of the human, but still be sensitive to what might be different in human. I mean, the nice thing is that some of the earliest steps in mammalian development are relatively well conserved, so we can sort of get away with that. And then, of course, as we get to later stages, then we can switch to pure human, because we'll be able to get enough data. And I also think at that point, there'll be enough cells where it'll have to diverge to organ-specific maps.
You know, I'd just like to point out at this point in time that the hub map people are constrained in that they can only work with human. They're not allowed to work with animal models at all.
That's why it's great that it's a bigger consortium then. I mean,
yes.
you know, but
Yes.
but that's what will, if you want to get development, and if you sort of want to get the earliest stages where a lot of important things happen, and a lot of insight can be gained, it's going to, unless you want to go into to serious ethics violations, you're going to have to do it in model organisms.
In terms of sources of tissue, okay, so I'm aware of the one in the UK, the one we have in France, one in Sweden. Are we missing like a big source somewhere in the world that can provide like samples? We can provide from 6 weeks, no problem, all the way to 17 weeks, more or less. So are we missing a source somewhere?
I believe there's an NIH-funded source in the US. I could dig out the details.
I believe it's in UW, and I believe they're trying to keep it relatively secret so that the people in the US won't realize that they have such a thing.
This is Kim. Can you hear me? This is Kim Aldinger, and I'm actually affiliated with the Birth Defects Research Laboratory, which is the human
there you go
resource.
I think we met before, right?
We did. We met.
I was about to dig your email while talking
to
this group. It was right before the COVID-19 broke out, so that's why I've been delayed.
And back in 2008, you know, there were some significant difficulties politically, and so it has sort of been a little bit quiet since then, but we're certainly still collecting samples and very interested to participate. And the 40 post-conception day samples, some of the really earlies, you know, they're 4 millimeters in length, so about equivalent to a mouse E10.5 embryo. So to give Dana some perspective and the rest of the group some perspective about size, and that, you know, profiling the whole embryo versus organs, I agree, would be necessary at those very early stages.
So what is the, when can you actually collect, the age range that you can collect, Kimberly?
So the majority of the samples we get are really mid-gestation. And so they're gonna be from 90 to, 90 post-conception days up until about 145 sometimes because of some discrepancy, you know, in dating. But we are collecting some earlies now, and the earliest that we've collected is 40 days. But those are more technically difficult to ascertain. So we don't have a lot, but we do have them and can ramp up. We've only been consenting 1 day a week, and there's an opportunity to consent patients 3 days a week for those early time points.
But sorry, did I hear right? That's 40 days. So that's still like 5 weeks, right?
Yeah, that's correct. Yep.
And Muzz and Alain, could you maybe share the windows of time where you most typically get samples, just so we have them across the other resources?
Yeah. For us, I would say easily from 6 to conception weeks to let's say 15, and that's what I mean. It's mainly we can get like 10 a week, okay, minimum.
So I think one of the, I don't know how everybody's sort of like using gestation weeks and post-conception weeks. Are we all happy with what they mean? Because it took me a while to figure it out. Post-conception weeks is -2 weeks from gestation weeks. Sorry, but it's +2 weeks from gestation weeks. So post-conception week is a bit older, if you see what I mean. So either we use gestation weeks or post-conception weeks. So 5 weeks, 5 gestation weeks is like, or the other way around, yeah, 5 post-conception weeks is 3 post-conception weeks. Yeah, it's -2. So just to kind of be consistent. So generally you get, from 4 weeks is probably like, you're then looking at 2 post-conception weeks onwards, which is really very, very young. And one of the things to highlight is like you can wait 2 years and only one of these samples comes from will be available from the perspective of like the HDBR as a resource. So I don't know what it's like for other resources and whether as a community, as soon as somebody gets one of these, then we make the most of that sample to get the best data possible, which, rather than to, and so maybe we should sort of like have some plans or outline of what would be the best thing to do and how to go about it.
I think one of the things that might come from this joint meeting is a way to have a common database of those samples that are available. And when a new sample becomes available, it is tagged within that database. I think this is true both for fetal as well as post-conceptional up through adult. If we can do that as a joint effort, I think that would be really useful.
Because the vast majority of the mid-gestational samples, that's very, very common, particularly from about 6 or 7 post-conception weeks up to about 12, 14 post-conception weeks for most of these resources. It's the ones that are at the very early stages and the ones that are at the kind of like later stages that's really rare.
I'm slightly aware of the time. Should we maybe, we've got a few things that we can do under this question. So setting up a database, maybe coming up with a common, with a wish list of assays that we might like to do if material becomes available. But would it be worth looking at some of the other questions?
Sounds fair. Yep.
Well, actually, I think we should try and wrap up with the conclusion of this one, because actually how we're going to get and potentially share and prioritize these samples, I think is something that we should at least have some working plan to resolve if we're not going to resolve it here.
I think sorry, Dana, my understanding was
part
right
2
now,
is for.
Exactly. Get the questions, okay, and then part 2, we get the answers.
Okay.
That's a good way, I don't know, but that's the...
All right, sorry, okay.
There will also be a session tomorrow talking about biospecimens and sharing them. So some of these points that we just brought up can be discussed there as well.
But related on this last topic, can I ask this, like if some of these samples are extremely precious and rare, and if they come up actually in one of the collections, is it possible to kind of coordinate to make sure the entirety of the sample is analyzed as opposed to a particular subset of cells?
I think that would be like the ultimate aim. If we can kind of like, come together as a community, that would be fantastic. That we somehow make that happen. And part 2 to solve it will be one way of how we go about it, really.
One thing we've done when we worked with tumor samples that were precious is that we had usually a plan up front of, for example, the order of priorities in terms of assays and how things would get distributed, so that it wasn't an ad hoc decision every time that we had a sample in question. And I think that helped a lot, not just streamline things, but actually end up with coherent data sets at the end, because not all samples always can work for all possible options, because they might be too small, or they might not exactly work, or you might not be able to run this assay on a sample like this or a sample like that. So it's nice to actually pre-think most of these things. It makes for better data sets at the end.
Just a thought on earlier gametogenesis. Much of the program is established in each of the gametogenesis for male and female. So there's a whole different gene expression profile and pattern that's probably very valuable to understand. Can we get at that? Should we get at that?
I suppose a lot of the in vitro fertilization clinics and male infertility clinics are already looking at single cell genomics of their samples and have lots of samples available.
It would also be helpful to those of us that collect materials to have feedback from those doing the analysis about what processing works best.
我刚和 NIH-HCA 的 PO 聊过了。你早，那个刚才试着这个语音不知道怎么弄坏了。我刚和 NIH-HCA 的 PO 聊过了。
I'm going to go ahead and mute that participant.
Shall we move to the second topic, subtopic? Okay. Which is how do we engage the developmental biology community?
I think we have to show them value. I think we have to demonstrate what they can get through working with us. We have to give them value.
So how do you think, how might we do this? I'm using the prompt questions. How might we demonstrate value?
So again, some of the recent publications that show that you can get insights from this type of data. Maybe some computational effort to give them tools, browsers, visualizers, something for them to interact with the data. I mean, again, all my work to date has been with mouse, but we did we got a lot of feedback from pure developmental biologists, again in the mouse world, just because they really enjoyed our data browser. So putting the data out there, but in a way that you can interact with it rather than core dump, I think is very empowering, when people can actually ask their questions and do something more sophisticated than a TSNE map.
So that's a great suggestion, Dan. And we had something like Annotaton as a community. So basically kind of like getting them to be more involved in the analysis process.
Yeah, but that's getting them to help us. We need to make tools that help them ask their questions. Maybe if they could see the value for them, and if we give them tools which help them, they might be more excited about helping us annotate.
And going back to what Donna said in the previous questions, there's a lot of value in being able to map between organisms that has been thus far kind of difficult to do, at least in humans, on later embryonic developmental stages. For example, we don't know exactly what the differences are between some mouse stages and human stages in certain organs, and that has a lot of value for developmental biologists.
Yeah, mapping across organisms is one of the computational projects problems that has recently taken my fancy.
And what about the forum to facilitate this? Because, how do we kind of actually offer them these tools and show them what value there is in, what the work that we're doing and how we will benefit them and how it might be of interest to them?
Well, there are two sides to it. There's an algorithmic problem, which is not solved, and I'm happy to participate with that. We're working on it in my lab, and I'm happy to work on it more collaboratively in a bigger group, as long as a bigger group includes nice and collegial and fun people. But the other problem is turning this type of thing into some type of interactive software tool. And here is where the type of things I like to do ends, so someone else would have to pick up that mantle.
There's also training.
Jonah, a question, like with the meetings that have been, that are starting to be organized, has there been good uptake from developmental biologists who are not necessarily familiar with single cell biology and the computational approaches? I mean, Gary?
Yeah, so about over 300 people signed up as interested for the summer conference that unfortunately now is going to be postponed, but and many looking through that list, there are many people from different developmental biology communities there. But I think we need to do more. So some people have got the message, like there's a cardiology community that seems to have signed up a number of people because I've had direct connection with them, cardiac development and our colleagues. But I think we need to do quite a bit more. So there's a number of ideas that came out of organizing that conference that we can put as action items later in the second part of this session.
That would be great, thanks.
I think also the Society for Developmental Biology has made a push to start to include more single cell biology and different folks from both the Human Cell Atlas and wider field. And so I think Samantha Morris and others are involved. So maybe also organizing and inviting them, but also making a concerted effort to go to some of those meetings and also some of the other larger international societies and development, I think would be a wonderful way to do some outreach.
I think training as well. So there's a big need from these communities to get people from those communities educated about single cell genomics technologies and analysis methods. And that's an opportunity for us to engage.
In LungMap, there's a real need to have a workplace and a playground. And the NIH has been chatting about a broader NHLBI broad playground where the wet lab folks can increasingly have access to the algorithms that more advanced computation scientists are using, just making it very user-friendly so that they can ask simple questions. Having training sessions is probably the most important thing, and subgroups with areas of interest. And then having young faculty bring their data to working groups that can analyze their data with various programs.
And for imaging, we do already training courses for imaging, and they're open completely. So we already had like 300 people attending them. So we're going to continue. So and we have a lot of people coming from the development community. But it could be better advertised or focused on development.
Or perhaps, Alan, that it's combined with a bit of analysis, if that would help.
Yeah, of course. We could think of organizing a workshop or something. It could be a good idea to have a workshop dedicated to technology for human development.
Is it worth trying to combine that with some kind of annotator-thon which taps the expertise of particular communities to identify cell types and to actually come up with some standards for annotation?
So one of the ways that I've thought about the annotate-thon would be you could just have like web portals, a bit like how Dana offered, and then they can kind of like do it online, that you can have it as like a meeting where people do it, or you can just have each one being given a link and they can just, fill in what they think is, what they feel. I think, Peter, you've done that, haven't you, Peter Kachenko?
Yeah, that's Peter's cell annotation platform. Maybe, yeah. At least I lost my Wi-Fi for a moment, but I see Peter.
Yeah. Yeah. Well, so we'll have an entire session on this tomorrow, basically in the afternoon. I think that there are a number of kind of parallel tracks there that people are trying to do, and for different levels. But this type of communal annotation certainly be something that we need to do, and I think at any chance we get. Right. So if we could set up an interface for people to do this, that would be great.
Yeah. Can I ask a question on this? And I apologize if this was just discussed. I lost my Wi-Fi, so it took me a moment to reconnect. But one thing I've noticed actually in this COVID-19 work, when we were going into the more and more fetal end of the spectrum, is that it becomes harder to map, right? You have the adult or even the newborn, and you have the 8.5 weeks post-conception. And the cell annotations, right, the tracking over time is a challenge, because they might not be as refined as they were before.
Absolutely.
So I'm wondering how this community thinks about this. Should we think about one annotation and another annotation, or the mapping between the annotations, or the levels? We didn't have a great answer for it there at all.
Yeah, I think unify those annotations are very important. Maybe first collect all the, breakdown based on the organs and age, development age, and then collect all the, at least all the published ones from the single cell, and remap them at, and we can have potential, temporarily put some of the name like a crossed ID or something, and then combine with all the development biologists and make the final mapping together.
Sorry to interrupt. I just wanted, I don't know if people noticed, the little pop-up should have popped on your browser that said 7 minutes, we're going to get pulled back to plenary. So I don't know if you want to try to tackle one more topic before we get pulled back.
So we should just say that, part 2, we want to discuss this further about the annotation across time. And then maybe should we jump to the pediatric one, Deanne, and because that's quite a big one.
Sure. We also might have an opportunity to talk about this during the second session if we break out. I don't know how people feel about that. Not that I don't want to talk about it. Okay. So pardon me for a minute. It seems
this is this the one that got combined in subtopic number 4?
Yes. Yeah, there we go.
8, yeah.
So there's a lot of relevance, obviously, to development of pediatric and adult health and disease, that you can go between development and pediatric data, because there's a lot of migration in the embryo of different cell types, and disruptions to those cell types migration may contribute to, for example, pediatric cancer development disorders. We have a list here of different discussion prompts. I don't know, I don't want to spend too much time on each of them because we're running out of time. But is there anything that anyone else wants to add to these and discuss? No, nobody's there.
The whole innate immune system and acquired immune system is being educated in the early pediatric window that has the long-term consequences for all health later. We're getting programmed then and we don't understand it very well. The trajectories of each of the immune cells and how they're interacting with parenchymal cells in each organ is going to be extraordinarily interesting and challenging in terms of analysis.
So I find that there are a lot of difficulties in how do you actually go about answering some of these questions. What are the tools that we have to answer those questions, both computationally and also experimentally? Because these are kind of like across the entire human lifespan. What are the best models to validate some of these findings and to actually prove these findings? Those have been, thoughts that I've had for quite a while.
In terms of pediatrics, there's also certain key developmental phases where certain birth defects are believed to arise. For example, on the heart, different migrations from the heart fields or the cardiac neural crest may have something to do with certain windows where developmental defects show up. And that may also be true for some pediatric cancers. So one of the things we probably need to do is engage experts in different organ systems for the most common birth defects. People like Sharon Plon or Philip Lupo or Wendy Chung, other developmental experts that are on the pediatric side, and try to find out from them where they studied that in mice. Coming back to what Donna was talking about earlier, there are mouse models for these developmental defects, but they're not perfect, right? And so it would be nice to have windows. How do we get these people involved so that we can start identifying the windows of development, where it would be great to start getting samples, prioritizing sample acquisition is one thing I would suggest for how do we.
Pediatric samples are canaries in the mine. In the strong phenotypes that are present in newborn period, for example, in the lung with severe developmental defects, really educate us about the entire programs for both later cancer and for cell lineages as the lung regenerates. So there's much to learn about the early progenitor and stem cells that are then reactivated during repair processes.
And in this sort of like transition from development to pediatric, we have like a big gap of the third trimester samples, which is probably like the prelude to the pediatric samples. How do we address this missing data, so to speak?
So
are there any resources? Sorry.
Oh, so I was just wondering, is it after the third trimester you're talking about, towards the end of the third trimester, or?
So most resources don't allow you to collect beyond sort of 20 post-conception weeks or 24 post-conception weeks. Thereafter it's kind of like, the fetus is viable, really. So we will have a gap in that sense of having material to profile those samples, and they are the ones that are kind of like immediately relevant to.
No, we can have some if it's a medical termination, then we have access to samples that are older.
The point is that the NICU has a lot of third trimester babies and they're not healthy, but they sometimes pass away there. In fact, probably frequently. So that's a potential source of tissue. And I guess the issue is that they're usually sick, but they might be sick in particular ways that affect one tissue and not another tissue.
But is there anywhere that has got like a kind of like a ethics already, or like a resource from those material, or you know?
I can follow up on that a little bit if you can hear me.
Yeah.
Yeah. So this is Gloria Prayer from the LungMap HTC. So we have been able to access some of these late trimester samples. Obviously, we're focused on lung, working through the donor network, the donor transplant network. So there is a neonatal donor network. Many of these are brain anomalies, intracranial anomalies, anencephaly being the most prominent. We are also able to take lung anomalies or lung disease. In some cases, those lungs look different. They look abnormal, maybe related to the anencephaly or to the congenital anomaly. In some cases, they may be immature or hypoplastic, but many of them actually look quite normal. And we won't know until we get down to the cellular level how normal or abnormal they really are. But so to answer the question, yes, there is a neonatal donor network. And there are research recovery organizations that will refer cases to you. There is a problem with changing attitudes within NICUs. And as you can imagine, it is very difficult for families, for parents. But many of those parents actually find some solace in making something reasonably good out of something quite awful in losing their child. So it is possible.
And could I ask, I know that the HDBR setup that we have in the UK here allows some of those samples to be sent abroad. Is there a reciprocal arrangement for some of these tissues within the US?
That is a good question that I think we should address in our ethics section here. I have, as part of the HTC, I have sent some samples, not live cells, but section paraffin, section frozen, and we have not had any trouble in sending those.
That's great to hear.
Mm-hmm.
Our challenge at CHOP for obtaining those samples, because we do have an early intervention center and we do get, of course, children in NICU, is just quality of samples, getting them through pathology and into the correct protocol. So while this is done at some centers, it's certainly always a challenge as well. That could probably be going into the ethics section. But how do you get these tissues and how do you get them rapidly into storage so that they could be basically not degraded by the time you're analyzing them? So we're actually trying to work on that kind of thing here, but it's challenging.
We had in AIM-1, I think, one of the kind of like putting all the repositories and tissue available. I mean, I guess this is one of those things that should go within the solutions, or how, what we propose as solutions within these discussions.
Correct.
I agree. I think having, even though this is not, something amazing, but having a good database that would list all the different samples and the ages that people can source, the ages of tissues, would already be a really helpful outcome from this session.
And would it also be something to aspire to try and make all these repositories standardized in terms of who might access the samples, what type of kind of like data access relating to the use of those samples, for the purposes of the kind of like HCA community, would that be helpful?
It would definitely be helpful to have the protocols associated with each sample published so that even if they can't be shared across continents, you know that there's somebody you could collaborate with, who those people would be. if you were going to do cross collaborations. And of course the protocol in which the sample can be released and used. I agree.
Yes, some of these discussions will again come up in one of the breakout sessions tomorrow on biospecimens and sharing of biospecimens. I hope people will join that, or we can join forces between these working groups.
Can I ask if for biospecimen sharing the developmental samples are particularly challenging? Is it more restrictive to share them? Is that the question?
From my standpoint, in the postnatal, right? So what I work with are all live born. I do not think that there is an ethical issue in sharing them, other than we need to be consistent with the consents or the authorizations under which the samples are acquired.
Because there's no blanket funding for obtaining samples where I am, a lot of these samples are collected under different protocols. So there's different restrictions, and that's where the complication comes in.
I think one of the restrictions too, or one of the complications, is how the data will be used, how it will be released. We talk about open access, right, versus controlled access. It comes down, you know, most of what we generate would be fine, but it's the potentially identifying nucleic acid sequence.
Yeah. I will say that the data coordination platform will have controlled access soon, so that should release that specific area of concern. Is that, and that my understanding that for pediatric samples, at least in the US, you would not be able to release without controlled access.
Yeah.
I do not mean from dysplasia, but from, they're a vulnerable population, so there's no way that you could do open access for that. But in any case, from an IT point of view, or from a data platform point of view, controlled access will be available similar to, you know, with the best practices, the same as a dbGaP or similar in other countries. So that should address that challenge, please.
So Gloria, can I just check with regards to the resource? Is it like you have a setup whereby samples are acquired at post-mortem and then they can be used for research purposes?
Yes, that's true. That's correct. Because I go through the transplant networks, there is in place protocols that shorten the warm ischemic time as much as possible. Again, it's a problem with the pediatric samples in particular because of the needs of the families. So it's not, you know, the couple of minutes, few minutes by protocol that it would be for a brain dead adult. So that does add some potential limitations to the quality. But some of the data that you saw earlier today on the preterm neonate 3-year-old, 9-year-old, that is data that we've acquired in this way. So the single cell is possible, single nuclear data with some caveats, but it's certainly better than access we've had before.
There's also a group I pasted in the chat called the Swifty Foundation, and they've been organizing post-mortem tissue collection from children. And we've been in contact with them, and we're actually working them as part of the CBTTC post-mortem tissue donation program. And so they're also another resource and may have some additional resources for us.
Thank you.
So I wanted to kind of like bring in some of our bullet points about, you know, the stages and kind of how we go about the analysis. And I think this relates to a point that Aviv made earlier about how you annotate based on, you know, gestational stage, because the cells are not quite equivalent. And we had some of the bullet points, which I think is a recurring theme within this, you know, this topic really, is with regards to kind of, you know, what are the kind of computational tools and how do we really kind of go about doing the analysis without, you know, without kind of like lumping everything together and then losing all the biology, because the cells are transient, they change, and so on and so forth. Any thoughts about analytical tools or how we might approach the analysis? I mean, Peter, you're here.
Yeah, I mean, I can chime in, but it's a tough topic, right? So I think Dana probably agree that, you know, trajectories are kind of key to development, right? So if we're talking about annotating even the cells, we basically need to think about transitions and their sequence of transitions, bifurcations, and so forth. And sometimes we see this in some data sets, you know, for some cells, and sometimes we don't. We only see like sort of patches, right, that are difficult to connect. But I think the developmental community in general has to be at the forefront of this as to how the annotations relate to sort of continuous structures, you know, both the bifurcations and the trajectories, and, you know, positions maybe of cells within them and so forth. I mean, technically, I think the analysis tools that are out there, they can capture these types of features in the data, but in terms of sort of annotating it and systematically approaching how to, you know, record it and name them, I don't think there's much there. So
I agree that we're really behind in annotations, but I disagree that our trajectory methods are sufficient. I mean, I think we have a good first shot, and there's a lot of good trajectory methods out there that have been very effective at discovering powerful and novel biology. But I think that we still can make them a lot more refined, particularly given that the data that's going to be coming from this sort of pediatric development world would be a lot more sparse. Sparsity is where these methods really fail. So when you have sparse patches and you don't really have all the continuous points, they can fail really miserably. So we have to think of ways to connect the dots also between time points and samples, especially since they're, you know, we'll get what we get, and to overcome sparsity. And I think that particularly for the goals of this atlas, much better methods that will really overcome the sparsity in this type of data really need to be developed.
So from the perspective of a solution, would it be helpful to kind of like take whatever data that's already there to try and see how we could engage the developmental biology to kind of like annotate what's there, and see whether it's refined, or, you know, using the data to see whether, you know, methods could be improved?
Well, I think you need a new language and a new, like, I think right now, you know, we're very good. I mean, we disagree on some of the names, we disagree on where to put the thresholds, but the field has very much been a discrete subset annotating group. So the field was into saying, you know, this is this stage. I mean, even let's talk about the thing I know, hematopoiesis, or even lymphoesis. So, you know, you have the double in B cells, you have all these stages, the double negative, the single negative, the this, the that, all the pro-B, pre-B, whatever, like tons of little populations that they've invented. But as a field, we haven't even developed a language on how to annotate trajectories. I mean, for me, I think it should be about trajectories and maybe sort of milestones along these trajectories, or places where something interesting happens. Again, going back to the B cell example, where I know like where VDJ recombination happens. So I would, you know, really want to develop a language, and I have no idea how to do that, so that we can actually annotate trajectories rather than subsets.
And I don't want to. I don't want to.
Can sorry. Go ahead.
I don't want to forget. I don't want us to forget that part of this developmental trajectories includes spatial. So not just spatial in terms of single cell, but also spatial in terms of the embryonic position and the common coordinate framework for embryos. Because a lot of these trajectories include cell migration. So not only do you have changes, molecular changes, but positional changes at the same time. And those positional changes aren't just in place, but they're moving across the axis of the embryo. So
I agree with this. Before we dig into the spatial one, I wanted to ask a question of Muzz and Dana and the others, which is if it's a reasonable assumption that profiling of human development is not going to be continuous. You're going to get a time point and another one. There's going to be a lot of constraints there.
Yes, exactly.
Just to say, is there value or help from model organism data, or is it mostly going to mislead us? So is it possible to, for example, chain between time points that in human we're getting too discretely by also seeing something more continuous in systems where we have more control, or is it actually going to deceive us and send us down the wrong path?
I strongly think that's a very good idea. I mean, that's what I suggested at the very, very opening of this thing, I think before you joined, that we're gonna, I mean,
yeah,
I particularly related it to the, I particularly related it to the very early time points where I know we're gonna sample very poorly. But yes, I think that because of our I mean, we need to do two things. One, we need to improve our computational methods so that they're better at handling this sparsity, because right now the current methods are extremely sensitive to this. And the second thing that we need to do is really try and fill in the gaps to the best we can with model organisms, which again requires developing methods to sort of do such an integration in a way that both allows them to fill the gaps, but also allows to account for differences between animals. And yes, I definitely think that model organisms in this particular case will play a very big and important role.
And do we have data to start? Sorry.
No, go ahead.
I was just saying that, you know, do we have a sufficient amount of data to begin to sort of look at some of these things now, you know, to see whether we can cross-species compare or, you know, start the kind of annotation based on trajectories, etc.
Has anyone ever compiled somewhere all the data that exists and that has been published in, you know, both, you know, I would say human, mouse, and primates?
I didn't
Knowing what data exists would be a great starting point, because I've seen a paper here, paper there, but I'm one of the most disorganized people on the planet. That's why I like working with Aviv. But I certainly don't have something like this.
I'm not sure what that actually meant, but
that you are so organized that I sometimes can't believe it.
So is this an outcome for us? Aviv, take it as a compliment, I think.
I would just not go down.
Yes. It was 100% a compliment, Aviv. I only compliment you.
Yeah, if there's one thing we've seen in this COVID-19 example, is that just by being forced to have to put a lot of data together, besides getting answers that are important about a critical disease, it also just forces you to grapple with the question of, well, I call this cell type and you call that cell type, and now we really just want to call it by something. It just makes people iterate through it much more quickly. So I would highly recommend it. And if I'm not standing still on this call, or Jeff, but where there was pediatric and fetal developmental data, it's of course in person as well. It's of course harder because the adults, even the newborn versus the development is difficult. It's not the same, and we don't have easy ways to change. I think it would also highlight where the gaps are, and that would also connect to what Deanne was saying about the spatial aspect and the fact that things are moving, and the sense of common coordinate frameworks for things that are constantly actually moving is going to be harder. It's going to be important to start thinking about that too.
So should we put in the blue boxes as an action item to put the data sets that are available in a kind of like a place where people can find it and actually start the process of, you know, if we were to re-annotate these data sets, what would the community now be calling these things, and try and sort of like see what are the model organisms data sets that are also available to put there.
I would say that besides putting the data out there, it's great to put the data with a person, meaning as you go into someone else's data and you're not even an expert in it necessarily, or you are, they have an opinion and you have an opinion, knowing who it is that you ask and come to terms around it. The community side of it is just as important. That's what the lung groups had, that they had this sense that they're coming together and they just iron out the details was very helpful.
Yeah, no, I think that's exactly, you know, where we have the data, and then where we want to use individuals' data, we have those people involved, but we also have like a broader community that can then, you know, input into the annotation of the data and how you interpret the data and so on and so forth. Maybe that's an action item that we can kind of start, you know, even now.
Yeah, so I've just, it's a bit impossible to keep hold of this, to, you know, record everything we're saying. So as the first output, I've just put interpreting the emerging development atlas, and I've put a couple of things down there. Maybe people can go to that first blue box and add things that they like, just so that, you know, we're catching some of the things that are being said.
Can I also mention that there's a lot of lower resolution data in terms of joining up animal and human models that already exist in the clinical literature and the developmental literature. So we may want to also provide that as a resource for people working in these groups. Like how mice match up with human at different developmental stages, things like that.
So how would we go about actually implementing a registry of all the data? Should we use an existing data portal, or is it just a spreadsheet that people add to?
I've been using mind maps recently. I'm not sure if it's useful. I've been using it for COVID-19. I don't know if anyone's ever used them, but I could show an example if anyone's interested.
Can you just describe what that is?
It's essentially a graph that you can organize by, it's usually an easy to use graph. Yeah, like topics or concepts, and then you can put papers on each one. Let's see here. I've got one that I can show you right now if you guys don't mind me sharing the screen, or do you want to talk about this at a later time?
Do you have a favorite mind map?
I've been using Coggle. I've been using
collaborative?
Yeah, it's collaborative. Let me show you really quick. Here's an example of COVID-19. So I'm, whoops, I accidentally moved it. So I have here molecular data sets in SARS-CoV-2, therapeutic research, clinical, and then I've got papers on the different things that you can link into, go to different preprints and stuff like that. And that seemed to help, but that this is just an option. Let me stop sharing.
I like, you know, the general concept, I think, of having registries for all the different data sets is actually really important. And it is, of course, possible for all of us to chip in and just do it, but actually having some central funding to underpin this would also be really helpful. So putting in a call to the funders, I think, would also be good.
Can I just check? We also have the DCP for the kind of like HCA related work.
Yeah, but I don't think the DCP is going to kick in with this in a timeline that's good for us, because I don't think that some of the non-human samples are going to be prioritized. And you know, I think we just need a list of where the data is, and by the time the DCP would have uploaded all that data, you know, it would be way beyond when we'd want to see that data, which would be in the nearer future.
Aviv, do you have any other organizational suggestions since you were
Sorry, I had to unmute myself. I think you captured the. Apparently, I've become the organizational liaison per Dana. I think you captured most of the tricks. I think people underestimate the amount of personal touch at making organizations like these actually the gelling actually happen. So it's never
we're not underestimating, which is why I think people raise the issue of funding.
It's not enough of just doing the organization that's needed in order to put all the data together and assign the people and so on. You need a couple of people who really, really care to, especially in the beginning, until it kind of gets traction, then they're not actually that needed anymore. So it's important to understand for a task like this, who's really the champion. Or champions. It just doesn't have to be a single person. But if it's everyone, then things don't tend to happen. If it's no one, things definitely don't happen. So there's kind of a magic number in between. That would be my additional organizational suggestion.
And this is a perfect thing to integrate trainees into.
Exactly. And again, if I'm just using the recent example that you guys have just seen in the, the handoff presentation, there were a couple of trainees that were particularly non-stop on top of this. And it's not because they were like the first author of the paper. It's a different dynamic when you do something like this. They might also end up being that, but that's not the point. That's not what motivates them.
So the kind of recurring theme that's coming through all our discussion is we wanted a kind of like a repository of what bioresources were available, what material people could get, what ethics, what kind of like data access policy, what data is already out there. So there seems to be this thing about a centralized resource for this community that's
yeah, it's like a sub-portal, and making the content of that sub-portal will be down to this community, but it can be built in a way under the bigger banner, because there are things that are very specialized to development and pediatrics, that you can't just do the typical things that the adult issue investigators do.
Because all of our discussion subtopics have come up with this as the recurring needs for the community.
So with one action item that we go back to our groups and ask any trainee in our group if anybody would be interested in volunteering to work with other trainees internationally to actually just develop a first version of this and see what happens.
Yeah, I might have a trainee that's interested in it, but I don't want to volunteer anyone without talking with them.
Yeah, we'd ask them, of course. And if no one volunteers, then I guess we're stuck.
I've got resources available, not trainees, but basically trained bioinformaticians that could help.
The other type of person who's good at this is database curators.
Yep. I have a question to kind of poll the group. So for the lung map, again, for the HTC, we have an inventory system. It's built on LabKey. It's quite easy actually to set up. Our bioinformatics support it. Do all the other consortia also have inventory systems like this, that their materials are available for access, at least within the consortia? Ours actually has kind of a shopping cart capability to it. Is that something that everyone has, or is it much more fragmented?
The liver team is using RedCap.
For us, we will have in France, it's going to open very soon, a website like the HDBR where you can log in and have access to everything that is in it. So it will be, of course, you have to get a login and password. We don't want everybody to access it, but then you can know exactly what we have, what is frozen, the type, a lot of information. And also you can apply for specific tissue or stages or whatever you want.
So that sounds very familiar, very similar to what we have as well. So maybe in some way, rather than reinventing and pulling them all into a common database, at least to have a portal that allows you to access each one of those inventories. Is that possible?
Yeah, I mean, if you go to the HCA website, there's already a development session where for now there's only the UK and the Swedish dental cell Atlas project. The other one, they are not, you cannot connect to it. But otherwise, it could be one way to access with this HCA portal, and then you can access every single initiative like this. I don't know.
So we have some funders here as well. Do you want to comment on anything? I guess that means you're just observing. I'm just looking at whether this is something that the community takes on a voluntary basis, or whether is this something that could be more structured, not just by volunteers.
I mean, sorry, this is Kat from Welcome. I'm observing, but I just wanted to say, I feel like this is potentially the kind of central coordination that HCA could think about. And I think as funders, we're definitely open to hearing what resources people need in order to make sure that this kind of coordination is happening.
Dan, maybe the one other thing I would add is, to the extent that this is being done by, individual members of the lab that are kind of going outside of and above and beyond what their normal projects or work would look like, some sort of incentive to kind of recognize that effort or help both give them recognition and also some sort of, acknowledgement of it is certainly something that we would think about and, helped try to provide in the past, for example, for submission to the DCP and other areas.
So I wonder whether on the subject of HCA coordination, there's a worry about the speed of stuff getting onto actually officially into the DCP and onto portals. But if there's a more federated approach that gets taken, there's still going to be some need for some standardization across those to make things cross-searchable. Is it worth talking to the DCP about whether using some basic form of their schema would provide a way to standardize even if data is not fully ingested? So at least the general metadata about experiment types and sample location and all of that will be somewhat standardized, and that could feed back into other sort of more central standardization efforts. Does that make some sense? Because obviously if everybody develops their own one, it's quite hard to federate.
So I want to distinguish between meta annotation and analysis, because meta annotation, if you didn't put it there at the time, it's sometimes hard to impossible to trace down. So if everyone could sort of take the meta annotation schema and try and stick to that, because I think the guys at EBI have made that public. That would be very helpful, because the more meta fields that you have about how that data, how and where the data was collected, the better, the easier it is to integrate at the end. Now, in terms of processing the data, I do think that developmental data might need slightly different processing than the standard pipeline. And even since I know that the person who's really heavy lifted and pretty much did, I would say, all the work for standardizing the sort of standard global data processing for all DCP HCA data, it's something that sort of works across tissues, across systems, across data sets, across technologies, and is like the best that you can globally. But even John himself, when he analyzes data in his lab, would not use that pipeline. So I think that we really should distinguish between how we process our data and how we annotate our data with meta fields. And these meta fields, if they're there to begin with, will allow for processing and reprocessing of the data however one needs for whatever format.
Very helpful kind of questions and feedback for the blue kind of boxes that we have. We have two more topics that we didn't discuss, and then we had the first subtopic which we didn't fill out the blue box. Do we want to discuss the other two topics, one of which was ethics around development work, and the other subtopic was the biology that we want to learn from the development atlas? Or do we want to go to the blue box, which is presenting the solutions from the first subtopic we discussed, which was basically looking at organ-based and anatomical unit-based atlasing efforts?
For the ethics, do you know if it will be addressed in the ethics session or not? There will be a specific, I think, ethic session.
Yeah. Are there any issues relating to kind of like policies by geography, specific regional constraints, GDPR? or do we think this is not a subtopic that's worth discussing? We can move on to the third one about the biology.
I mean, I think with the ethics, in many ways, we've already highlighted some of the problems that different groups will be able to access their tissue samples under very different ethics, whatever was appropriate for the study at the time, and that will obviously limit the ability to share tissues to other studies. So I think making some efforts going forward to make ethics as broad as possible would be really helpful.
So by this you mean kind of like making the repositories have similar ethics?
Yes, yes.
Is this kind of achievable, or is this, what do people think about the value of pursuing this as a focus group outcome and solution?
It'd be nice to know if there were specific challenges that people are facing with ethics. Like I see a lot of people accessing samples already from HDBR and other things. So the ethics approval, it seems to be occurring, I presume, smoothly. Is that not the case? Or are there specific ethical challenges that have come up in existing work?
No, I think it's more to do with like some of the kind of like, for example, the older samples that Gloria was alluding to, the access to that and the ethics associated with it, which is different to the ones that are kind of like HDBR related, which may be different to the French Hugo Decker or, Hugh Decker, sorry, those sort of things. Is there a need to standardize, or do we just kind of like, is that too much to take on, really?
I think it's difficult to standardize the ethics if you're talking about, every donor or every source of tissue would be guided by the same, we have the same ethical principles, but some are consented or authorized for different uses, right? And we have to be consistent with how the tissue was originally gotten and for what purpose. But we've tried to address that by putting in place data use agreements or material transfer agreements, and not so much for the protection of the data, other than the protection of the human privacy and confidentiality. So that's been the best solution that we've been able to come up with. Is that investigators are responsible for not re-identifying individuals, for example, and I don't know a better way than that. I'd be happy to hear it. If that's the standardized approach that people are trying to identify, that would be my suggestion. That's one of the standardized approaches.
No, I think standardizing ethics is going to be really practically very, very difficult, and probably hitting our heads against the brick wall or something.
Yeah.
So it might be that we need to kind of follow your solution and actually maybe identify partners that are.
Oh, you've frozen.
Benefit of this consortium. Am I frozen? Sorry.
No, you're back. I'm sorry.
Sorry.
That might have been me.
I said that it might be difficult to actually standardize ethics, and I think what you suggested, Gloria, is like a more pragmatic approach, perhaps. And what we need to do is just identify partners who are, willing to kind of like work together for this consortium effort.
Yeah, I would also say that perhaps within these databases, these inventory-like databases, that one of the metadata fields is, how is the material consented? What is it available for? Does it need to be controlled? Does it access? Is it available for open access, etc.? Great.
What about the section that we didn't discuss earlier, which is biology from the Development Atlas? Is there appetite to kind of discuss some of these discussion prompts that we filled out? Particularly the temporal dimension.
I wonder a little bit, I mean, clearly there's an enormous amount of biology that we hope to learn from all of that. I'm just wondering whether within the context of what we're doing here, I mean, one of the remits that we've been given is to come up with some specific suggestions, whether it might be worth revisiting some of the things that we did discuss earlier, where we just raised questions, but where people did seem to have answers, and just sort of fill in the missing gaps there. But it's just a suggestion. So for example,
I'm sorry. I actually do think that I'd like some of the, particularly the real developmental biologists on this call to say, what are the most important questions they'd want answered from the atlas? Because I think that, you know, we can collect data till tomorrow, we can coordinate this collection, but as any scientific project, you have to have priorities, you have to have trade-offs. Priorities in what you collect and what samples you put your money on, what assays you apply to these limited samples, and trade-offs in any computational method on the planet. It will have trade-offs, and that really needs to be driven by what's more important to get out of it. And, you know, I can you know, be a couch developmental biologist and invent my own questions, but I would really love to hear what, you know, the card-carrying developmental biologist and a pediatric biologist would most want to get out of such an effort.
It's something we discussed in Barcelona for the HCA meeting. We said, okay, what would be the questions that developmental biologists would like to address in their own system? Could we identify key questions for various organs that are development related and that we'd like to answer? Try to stimulate like discussion between people in this field and say, okay, this is what is for us a key question in human development, and then see how people that do technology could help answering those questions. So as you said, first identifying the questions, and then trying to see how people in the consortium or doing a single cell or imaging can help answering those questions. So I'm trying to gather people to try to motivate them to list questions would be a good thing.
No, I agree, because I think, as Dana said earlier on, you know, what can we do for the developmental biology community? And this is one of the things that we can say. These are the questions that we can answer with the technology and consortium effort that we have. And therefore, it would also help us with the second subtopic in terms of how to engage with them.
So I agree, and I think the question should be focused on the technologies that we have available. So I like the question that we were discussing earlier about how to define cells and trajectories. Because that's really an area where we could maybe add, advance the field of developmental biology by defining these trajectories and coming up with this language that Dana mentioned. I think in that we need to have defined, you know, states. Like if you're annotating the trajectories, it's not just single annotations; it's multiple overlapping things. Like you might have, just as an example of how to answer this question, new concepts that might come up. You might have transcriptional programs that are pushing a cell in one direction or another, and they're overlapping each other. And those transcriptional programs can be defined based on where the cells came from, spatiotemporally, and also where they're going, spatiotemporally. And that's not something that's something that a lot of developmental biologists think about a lot, but maybe not in that computational or formalized language type of way. So I like that question.
The question is also kind of quite complicated in the sense that it depends on the gestational stage as well, in terms of this is the whole temporal aspect of it. Like some tools are going to be so pertinent for some stages and other tools are more relevant for other stages. It's such a dynamic, like a fluid system, or, you know, it's quite complex, I suppose.
I mean, there are millions of infinite questions and people have been answering them anyway, right, without us. So, you know, I think we should try to focus on what new things can be done now.
So how do we do this?
I think earlier on, when we were having our moderator meetings, I mentioned that it's sometimes a little difficult, or maybe it was another meeting, but sometimes really difficult to engage the developmental biologists and the pediatric people I think one of our to-dos would probably, how do we do this outreach to get this feedback? I know that's really basic.
I think collaborations is a good idea. So I know I've been learning a huge amount over the past few years by collaborating with developmental biologists, and I'm sure Dana can say the same and other people can say the same. And really kind of part of it is anthropology. It's studying what the developmental biologist community wants, right? And what they need and working with them, right? That's frequently computational biologists do that. So that's one specific area that we could provide our, what we've learned from these individual collaborations. State thing that I was telling you about is directly related to a project I'm working on with a developmental biologist.
So would it be helpful at a kind of like a overarching level for HCA or HubMap to kind of like interact directly with the Society for Developmental Biology, which Jonah suggested, and say, what would you see as kind of like critical questions within human development that you would like answered, or where are our kind of like a common shared ideas and goals?
I mean, I'm happy to go to the next meeting and try and do this type of outreach if and when that meeting will ever happen in these current days.
So we can put that down as recommendations of what we want to do, I suppose.
Yeah, it would be great if we could maybe talk to the organizers of that conference to have a session about HCA at that meeting. Maybe that would be great, and multiple speakers can participate.
Another good venue might be the American Society for Human Genetics meeting. There are a lot of developmental people in terms of, genetics of birth defects and things like that show up at that meeting. I know that Gabriela Miller has shown up at that meeting. Even though it's a genetics focused project, there are a lot of genomics people as well, and there were single cell meetings last year in the conference, and they do support breakout sessions.
So which blue box is this going into? Should I start one?
I think it's gone into the blue box for number 3.
Yeah, that's where I've started filling out actually. I shouldn't be scribing. I'm sorry. I kind of.
Yeah, no, please do. I mean, I've put a couple of things, but I'm happy for people to just jump in and put more things. I'm sure Jonah will be the same.
May I make a process clarification? So I'm going in and just getting some of the details, like who's scribing and who the moderators are, and I'm copying that for all the boxes since we're doing it all together. So the hope would be that each one of these blue boxes stands alone as something that might be taken on by members of the various consortia. So I just wanted to make sure that each one of them was kind of just addressing a single problem, that up front we identified which problem we're addressing. Then the notes section seems to be pretty good here with the proposed solution, and that they were kind of one per blue box.
So who should be filling out the blue box, Stavros? Sorry.
The scribes, right? So so for example, if I just look at the very first one, it says, somebody's pasting things. I'm jumping around. The problem we think we can solve by collaborating. And so you have your recommendations, trajectory analysis efforts, but we need a problem in here. So what are all these ideas listed below? What are they addressing? Which one of the questions from above?
So we've kind of done it slightly back to front. One is the one that we haven't done the blue box for.
Okay.
I think we've done the blue box for 2, 4, and 3, and then we were going to go to 1.
Okay, so for 2, looking at 2, problem we think we can solve by collaborating, recommendations. Actually, I'm probably 2 is empty.
Yeah,
3 does have information. Yes, got it. Okay, so you guys are working your way, and I will go in and populate where it says moderators, note takers, I'll put all that information in.
So subtopic 4 is also where we filled out the blue box. So maybe we should finish off the blue box for 3 and move to 2 and 1.
That'd be fantastic. Thank you.
Anything else we want to add to 3? Sorry.
Yeah, just who would, can we identify an action item here? So I think it would be great to engage these sessions, these meetings. Does anyone have any specific contacts on the organizing committees of these meetings that we could send an email to and ask if maybe the HCA should, I don't know who would be appropriate to send such an email, but we could develop a quick plan.
I mean, given that we now have the networks, the biological networks, I think the leaders or coordinators for each of those networks would be very appropriate to send those emails out.
Okay, we can do that. I think for development is Deanne, Stan, and myself, so we can start doing this. That could be like a direct action item with names held responsible for delivering.
Great idea.
If it's helpful, I can provide contact information for the American Society for Developmental Biology, with some of the organizers and also then the staff.
That'd be brilliant.
Okay, no problem.
Shall we go to number 2 now, from 3? Which, oh yeah, this is very similar actually, engaging developmental biology community. So can action items for 3 also, you know, be repurposed for 2? I mean, or we blend the 2 together, the action items for 2 and 3 together?
Yeah, I'm sure we can do that.
Which leaves us with subtopic 1. And this was our first discussion.
I pasted some notes of mine into, I know it's the right thing to do, but into the green section. That kind of summarized the discussion on databases and being able to search for stuff.
Yeah.
I can move it down.
And as people were talking about developing repositories, I wrote a couple of notes, but we don't really have quite the right title for this question. So we haven't really defined the question, but we have really talked extensively about developing repositories as a resource for the community as a whole.
One thing we discussed earlier was having easy to use online systems to help the developmental biologists. Is that something that is related to this? That's more concrete? I'm not sure.
In topic 1, you mean?
Yeah.
Because it's true that a lot of developmental biology, at least the people on the pediatric side, really focus in on organs. So a lot of the use cases and collaborations are probably going to be organ-based, at least on pediatric side.
Do we want to say anything about the CCF in relation, or Common Coordinate Framework in relation to development? Is this, how do we go about this sort of, how do we achieve this?
Is there a Common Coordinate Framework network or group of people that I guess would be useful to link the developmental biology network that Kirsten mentioned and you guys are on, with a CCF network if it exists, or who is working in it?
How would even a common coordinate framework work here? Because the common coordinate framework, to my understanding, and Aviv can correct me because I think she's a lot more involved in it, is you have an adult organ and there's human to human variability, but you want to sort of be able to map them. But the whole idea about development is that the organ in the earlier stages, first of all, undergoes tremendous morphogenesis, depending on what organ, how far, and changes in such a way that I think one of the things that makes a common coordinate framework for developmental biology so challenging is the dramatic change in what that coordinate framework needs to be over time. And so, I'm wondering how that would even work when the thing itself, when the framework itself is constantly changing over time. That's what makes us special.
Exactly. So how do we kind of like approach?
Doc, what are you eating?
I think when I was discussing this with Alain a while ago, mentioned that the embryology field already has a lot of these frameworks that exist. So it seems like people are very knowledgeable about those existing frameworks should be participating. And then secondly, the people in the HCA thinking that common coordinate framework should be linked somehow to a developmental or pediatric project such that they can address the changing nature of it.
So would it be worth in engaging the kind of like developmental biologists with regards to the people from Society for Developmental Biology or Human Genetics, that we present them with not only the biological questions that we want to, that would be kind of like a common theme to bridge us, but also what are the kind of like a basis for the common coordinate framework that they've already got established that we can then leverage and deploy or adapt with the data sets that we have.
I think it would be great to really get from them their naming schemes for things. I myself have only looked at really, really early stuff, so I know naming schemes well earlier than most of what we'll be dealing with here. But I do think that whatever naming schemes currently exist, they'll be insufficient, and I think it will take a while for the common coordinate framework to even figure out how they're going to handle the adult. And in the interim, my attitude is to take pictures of where you're taking the sample from at different magnifications and resolutions as much as possible. Because again, even if we don't have names for things now, with images of where the sample came from, we can fix that later on. So anytime it's possible to image where you're taking the sample from, that will be enormously powerful as these names for things and as the coordinate, the coordinate framework solidifies over time, because we're collecting the data now.
There are a lot of physiological marks, coordinate marks that the biologists use that are studying things like whole organs that already exist. And it would be great. One of the things we could offer the developmental biologists in these fields when we get them involved is mapping what Donna was just talking about directly to the old-fashioned coordinate frameworks they already use. There are a lot of medical names and terminologies for different, like the second heart fields, the cardiac neural crest, and people have some detailed ideas because they can do staining in fish of how things move around in the embryo. And that would be a big, I know we talked about this already, but that would definitely be a big win for us to try to get that, that connected for them.
I agree, and sorry.
If you think about it, the anatomists already have an enormous nomenclature for macroscopic points in the body. I mean, the common coordinate framework is going to be sort of like that picture that you see in Gray's Anatomy. Initially, it will be whoever gets there first creates the sort of initial framework, and then that's refined as other people come in and do more things. There's a lot of talk within the hub map about using the vasculature as a framework. I mean, the major blood vessels have all been not only mapped by anatomists, but positioned. I mean, there are 3D models of what the major blood vessels are. Most of the major blood vessels will penetrate a lot of the tissue samples that you have. I mean, they can be used to create an initial framework. you know,
no, I agree.
distance-wise.
It's very important that I think we actually use what's already been established, because there's no point in reinventing another parallel layer of science or nomenclature. And it makes, like, you know, it puts historical kind of like nomenclature, we leverage that so that we can actually put the new sort of like data sets in the context of existing nomenclature. So I think, you know, these are great suggestions.
Just a mid note, I added another blue box underneath this blue box just to handle the CCF.
Great. There was one other thing as well that I didn't think we discussed. What do we think about, there are a lot of groups who have generated different data sets, and in a way that kind of like was highlighted with the COVID-19, how everyone came together with kind of like old data sets and new data sets and analyzed it as a kind of like for a bigger picture as a community. This might help kind of like boost numbers and actually be able to answer some of the questions about, you know, male-female differences and things like that. Would this be helpful?
Definitely.
So should we put that as an action item, that we build layers of analysis or kind of reinterpretation as a community?
If I gathered your question correctly, you're suggesting that's part of the analysis plan, right? It would be great to have an analysis plan. What are our priorities to figure out what are the, you know, what's our scaffold that we're all going to be working toward completing?
Because we would have all generated like data sets, but there's then the ability to pool all the data sets and kind of get a better understanding, because with all our individual data sets, it's quite hard to understand male and female differences or whatever. Whereas if we pool the data sets, we might have the power to do that.
Right.
In the way that the COVID-19 research, I thought, showed a lot more, you know, insightful biology than if it had been approached as individual kind of studies.
Yeah, coalesced and pointed.
One last thing we should do here, we talked about it at the very start, about if we got a precious rare sample, what would be our ideal kind of experimental plan? And we thought we should kind of briefly outline it.
So one suggestion I would make to always have, whenever we have single cell, that there's also some spatial component so that we can start to link the two together. So that when we preserve tissue, we always have one sample that might go to a single cell analysis and then one sample that might go into a spatial analysis.
I think epigenetic maps would be particularly useful for development.
I think there will be also a discussion about optimizing and maybe combining using the same samples with various techniques. So it's also something to work on. Can we do, I mean, how much can we do with one single sample? So that's something we're trying to do in the FRESH consortium, trying to see how many techniques can we combine with a single sample. So can we process with one, then process with something else, and process with something else. So it's something that could be useful.
There are a lot of new methods that try to combine multiple genomic layers in the same method, right? And it's so like papers every few weeks about that. I guess this would be a really good use case for those. Is that what you were talking about, Alain?
Yeah, not only genomics, also like what we do for imaging, spatial transcriptomics together with other things. Can we then dissociate the cells, do something else with the nuclei, etc.? So can we do a first round of imaging, then still use a sample of something else? So it's something we're testing.
The slight kind of like logistical issue is there's like a lot of projects that get submitted to all these repositories, and then you're put in a queue. And I think it's very difficult to kind of be able to justify that, you know, the consortium can work on the next, you know, available sample or whatever. It's, you know.
I agree, but also because we're not also connected. So I don't know what are the projects that you receive at HDBR. You won't know what are the projects I'm going to receive in France. So at some point, it's also something we discuss. I mean, maybe there are 3 groups working exact same thing in the world. So maybe we don't need to do the job 3 times. So if we could have something where it's more like integrated between us, or we have more discussion, or try to avoid like duplication of the work, that would be also good.
Yeah, that's a great suggestion. Actually, maybe we should inform the resources that we are kind of like working with to say that we would partner with, you know, whoever else who've also registered a project for the same thing to kind of like work together. That might be helpful.
Yeah, or something where people, when they ask for samples, they apply at the same time to a common portal, and then it's between us, we decide who's going to give them the tissue, etc. So it could be also one way to coordinate better.
Mm-hmm. I think that will also go down well with the repositories, actually.
Is that another one of them? You already gave me 2. What would be not?
Looks like we only have a few minutes left. Do we need to go over any of the blue boxes to attach names or anything else?
That sounds like a good idea if we just kind of like recap and then see if there's anything else to be added.
You guys mind starting maybe from the top, starting with number 1, just kind of going through it, doing a check?
Can we please mute the noisy?
Yeah, I'll find out who it is. It looks like it's Betsy. Sorry, it's going to take me a minute. I have to claim host. But while I do that, maybe you guys can start with the first one and just have a look at problem we think we can solve by collaborating, put some text in there, and then make sure you're on the same page about recommendations, make sure I've got the names right in the next section, and if there's any other expertise you feel you need. It'd be helpful if I shared my screen, or can everybody see what I'm talking about? And unless you have a next meeting scheduled, then I don't think you need to put anything there. So.
Yeah, maybe you could share your screen, no?
Yeah, sure. So here's the document we're all looking at. Up top is where I put the table of contents. So subtopic 1 is the first one you discussed. Here's the yellow or green section that you filled out originally. And then here's the first blue box. And so it says title, Interpreting the Emergent Development Atlas, and then problems we think we can solve. Yep, problems is fine. Then these are the recommendations that you put in. Thank you to whoever's typing this in. I cut and paste some names here just from the information I had. So just making sure that that's correct and accurate, that would be grateful. And then once you guys have filled out, if you're on this call and you don't see your name, but you've been an active participant, please just add yourself, and then I'll cut and paste this section down to the rest. I'll save you that busy work. And then if there's any expertise you need, if there's any community you need to hear from to kind of make this more realistic, I heard at least one person call out a specific community earlier, yep. And that would be helpful. And if there is a next meeting, which I haven't heard any talk of a next meeting, so I think it's fine to leave that last section blank. And then if all 5 boxes look like this, or if you need an extra box for anything, if it's actually 2 ideas, that's what we'll transfer over to the gallery at the end of the day.